Okay, but one mushroom is a huge amount of food for one of these mushroom fly larvae because they're very small. So you normally have males and females. They find a mushroom and lay eggs on it. And the larvae comes out, and it starts to eat the mushroom, but there's plenty of mushroom around, okay? And one larvae would take a long, long time to eat that whole mushroom. So what it does is it actually starts to produce offspring inside of itself, okay? It undergoes parthenogenesis. It doesn't lay the eggs. The offspring just develop inside, and then they eat their way out, okay? But that means that the female has like 100 offspring. This larvae has like 100 offspring inside of it, and then they just eat their way out. And they keep doing that until they have this big population that can consume the mushroom, right? And then when the mushroom starts to run out, then they're like, oh, it's time to get out of Dodge. And so then they stop doing that. They go ahead and form pupae and form adult males and females, and then they can fly off and find another mushroom, okay? And so we can actually force other animals to do this in strange ways. So this is an example of C. elegans, okay? So remember, C. elegans females normally have sperm and egg. And they fertilize, but then they release the eggs, okay? But we can mutate the C. elegans so it can't release the eggs, okay? And we can do this genetically so that all the progeny of that do the same thing. And so this is a little movie I hope it runs, yeah. And so if you see this animal, those are living worms inside of it, okay? So it's a hermaphrodite C. elegans, and then those are all its embryos that have developed, and they're going to eat their way out, okay? But then they are going to suffer the exact same fate. They are hermaphrodites. Their offspring will be inside of them and then eat their way out, okay? So it actually, and so this is an unusual situation for C. elegans, but some animals really do this as a way to propagate, okay? And then if conditions change, they can revert to sexual development. What's that? It'd be dead. Yeah. But see, it's gone from 1 to 100, even though it itself has been sacrificed, but it's increased its number substantially. Okay, and this is called a bag of worms phenotype. Okay, and then there's some other unusual examples. So people usually think that if you're a multicellular organism, right, an animal, large multicellular animal, or any size multicellular animal, that asexual reproduction is actually a dead end, and that fits along with the thinking that really you need it for recombination, for genetic diversity. And so the thought is that you transiently will produce animals that are asexual. So you can have like those New Mexico whiptail lizards. But the thought is that they're short experiments in evolution. They're not actually going to make it very long. Okay, and we mean they're not going to make it for a million years or something like that, right? Which is a very short period of time for a species. But there are a couple examples that people have become very curious about. One of these is this thing called a Deloid rotifer. Okay, so rotifers, very strange looking small animals. Nobody knows exactly what they're most closely related to. They're probably most closely related to arthropods in some way, but distant. And these are some pictures of one. And it turns out that they've gone 80 million years without having sex. Okay, so they just reproduce parthenogenetically. But it seems very odd because by most models, you just couldn't survive that long as a species if you didn't have sexual reproduction. So they reproduce exclusively by parthenogenesis, and they are clones of each other. Okay, so it turns out though that it's possible. There's a recent paper last year that says, well, maybe there's a solution to this. The reason that they have genetic diversity is they're actually very good at absorbing DNA from the environment and finding genes, okay, from organisms that have died, but their DNA is still intact. And in fact, it turns out that it looks like if you look at rotifers, about 10% of their genes have come from somewhere else. So they've actually absorbed them into them, okay, and used them. And so some of the biochemistry that they're able to carry out are not genes that evolved in them, but they're genes that they've actually taken from other microorganisms and other multicellular organisms and adopted. And in a way, this is actually also the solution that bacteria and archaea have come to. So we talk that bacteria and archaea only reproduce by cell division, okay, but they can actually undergo a type of mating. They just don't have male and female sexes the way we think about it. But they have mechanisms. For example, they can form a structure that allows small molecules of DNA to replicate and pass between them. And we also talked about, remember early on, we talked an example about where you could heat kill a bacteria that is pathogenic and mix it with another bacterium, and that bacterium would now become pathogenic. And what that bacteria was taking up was DNA. So the pathogenic bacteria had been destroyed, but they still had fragments of its DNA. Some of those fragments could be absorbed by the non-pathogenic bacteria. Those genes, yeah. So it can be simply random, and then selection acts to keep the ones that have some function, right? So a very good example of that is antibiotic resistance. So it turns out that, I think we might have mentioned this, right, that there are many soil bacteria that actually do have genes for antibiotic resistance, and they have them because the antibiotics are produced normally by fungi that are in the soil. So a bacteria that normally attacks you has no use for that antibiotic resistance because you're not taking that antibiotic resistance 1,000 years ago. Now you are. So now if that gene gets taken up, there's an advantage to having that gene, and then those bacteria live and propagate. Okay? So that's the idea, is that you have a way to increase genetic diversity. It's not by mating, but here's an alternative. You can just grab genes from other organisms at random and test them and see if they're good for you. Okay? Again, you don't want to anthropomorphize too much. You're not really doing anything like that. It's just selection. Okay, and so bacteria have a number of ways of doing this. And it looks like essentially Deloid rotifers, even though they're multicellular animals, have adopted a scheme like this, and that's how they've managed to survive. Okay, so now we're going to switch gears a little bit and talk about what happens, though, when mistakes get made. And again, the idea here is that these mistakes are pretty informative. And what happens sometimes is that a chromosome gets lost during meiosis. And what we mean by lost is that it undergoes this event of non-disjunction. So it doesn't get segregated properly. Okay, so they line up at metaphase, and then when they get pulled apart, right, in this case, for example, the two homologs are supposed to go to opposite poles, but they don't. And so two go one way, and nothing goes the other way, right? And so then you'll end up with this kind of product, and this is just a cell with two chromosomes, two different chromosomes here, right? Simplify the drawing. And then you'll end up with this situation where there's some of the products of meiosis have an extra copy, and other products of meiosis are missing that copy. And this non-disjunction can either occur in meiosis 1 or meiosis 2, okay? And that just changes a little bit what's going on. So if it occurs non-disjunction initially, then you'll get two abnormal gametes that have extra chromosomes and two abnormal ones that don't. And then if it occurs later, you can get different patterns. Okay, and this just, if you follow this, you can see the kinds of things that could happen. And both of these things happen in meiosis, that you can get a non-disjunction in either of those. But what it's going to mean is that it's going to give you the possibility of creating offspring that instead of having two copies of a chromosome, either have 1 or 3. Okay, so if one of the gametes has the normal 1 copy, but the other gamete has 0 or 2, then the diploid offspring is going to have the wrong number. Okay, and so this happens, right? And these are called trisomies or monosomies. Okay, so having 3 copies of one chromosome or 1 copy. Okay, and for most genes, okay, so your average gene in a human genome, having 1 copy, 2 copies, or 3 copies is fine. It doesn't actually matter in terms of overall viability. Okay, it doesn't really give you a phenotype. But there are a few genes where that's not the case. Okay, and in those cases, either you have to have 2 copies. If you have 1 copy, you die. If you have 3 copies, you die. Or you don't die, but something goes wrong. Okay, so if you have trisomy or monosomy of an entire human chromosome, okay, there are phenotypes as a single chromosome contains hundreds of genes. What that means is that, okay, so your average gene, it doesn't matter. But if you've got several hundred genes, there's a good chance on a chromosome there is a gene there where it does matter whether you have 1 or 3, even if you don't have 2 copies. So in fact, for humans, if you have monosomy for any one chromosome, it's not viable. Okay? So on every chromosome, there's at least one gene that requires 2 copies. Okay? But trisomy sometimes happens. Okay? And this requirement of 2 copies is called haploinsufficiency, that you need to have, that being haploid for a gene is not good enough. You need to have 2 copies for the organism. Obviously, the gamete's fine, but it's the zygote that has a problem. Okay? So some trisomies in humans can survive, and right now we're just going to talk about humans. Okay? And that means survive to birth. So many trisomies probably occur, and you never see them because the embryo ceases development right away. In fact, it might cease development even before it can get to 2 cells. So it's, it's not going anywhere. But in a few cases, you can actually get to birth or close to birth. And so these are the chromosomes where you can have trisomies where you can recognize that they can make it through part of development at least. Okay? And some can be viable humans. All right? You'll notice that it tends to be all of the larger numbers that are survived as trisomies. Does anyone have an idea why that is? Yeah, they're smaller, so just by chance, they're less likely to contain a gene that's going to kill it, right? And the most common one that can become adults are trisomy 21 or Down syndrome, okay? And so this is a situation in which there's an extra copy of the 23rd chromosome, okay? And there are a number of phenotypes that result. There's nothing, other than that stunted growth, there's nothing that's absolute, and severity can vary incredibly. So for example, the mental retardation. There are some people who clearly are Down syndrome's patients and have this trisomy 23 that you can't tell that they are in terms of like mental abilities. And there are others that are severely affected because there are a lot of interacting loci, okay? But there is an effect. The reason that the other trisomies don't occur very often is because they usually aren't viable, okay? And even the other ones that were listed are very severe, and most of the offspring actually don't survive very long because they have much more severe defects. What happens on the sex chromosome? So this could also happen to X and Y, okay? And so this does happen, and so you can get these triple X or XXY or XYY, okay? You can think about what the gametes are to create those, but they happen. So triple X are essentially normal women, okay? There's actually no obvious way to identify them, okay? But they do occur, okay? XXY is Klinefelter syndrome. This one has a clear phenotype. So these are men. They're men because they have a Y chromosome, so they have that SRY gene, makes them anatomically men. But they have reduced levels of testosterone, and they're infertile or reduced fertility. Okay? So this one is usually noticed. Okay? And they also have some phenotypes. Okay? So for example, they actually end up taller than most people. Okay? And it's not known exactly why that happens, but that is part of the phenotype as well. And then there are XYY, so men that have 2 Y chromosomes. And this occurs about 1 in 1,000 men. And there's no obvious phenotype. There's lots of things in the literature about, oh, well, no, they're more violent and they have, aggressive tendencies. And it's not clear that any of that's true. And most men are never going to know that they're XYY. Okay? And they're normally fertile and everything. So chances are that there really is no phenotypic effect. Yeah. Right, so, because you package a sperm with 2 Y chromosomes in it. Does that make sense? Okay? Yeah. Right, so what will happen is that you actually still can get meiosis to occur properly in these, okay? So, but it's a problem here, not actually in meiosis, but it's a problem in actually making sperm at all properly, okay? Yeah. That's a good question. Let's see. I'll have to think about that, right? I don't think so, because it's not known as a phenotype, but I'll think about it, and you can think about that. Yeah, let's see, but I think we have to try it out and see. That's a good question. I'll try to look it up and see for you. Okay. Okay, so let's move along a little. Okay, but what happens in a species that has chromosomal sex determination? So the sex is determined by the number of X chromosomes, such as in Drosophila. Okay. So it's not the Y, but it's the number of X chromosomes. Okay. So in this case, XO flies are male, but they're sterile. Okay. XYY flies are female, and they are fertile, and XYY flies are male. Okay. So again, that's because it's the number of X chromosomes that sets it. All right. So non-disjunction can also occur during mitosis. So we talked about that in meiosis. Okay. Usually when non-disjunction occurs, the cell will die. And we'll actually go through at some point next week probably and explain to you why this happens. It's a way to prevent you from having a serious problem. And this is one way that you get cancer, is to have a cell that undergoes non-disjunction and doesn't die. Okay. But if it happens to a sex chromosome in Drosophila, you can get an individual that's mosaic. Okay. It's not going to kill it. So you're going to get this non-disjunction in mitosis. So that means you're going to have a cell in a female fly. When it divides, there's going to be an error. Okay. And what you can actually get sometimes is, so normally you would just get XX and XY. All the cells would be identical. They'd all have 2 copies of the X. So what you can get, though, sometimes is an XO cell to be made. Now, you might think, well, this should be triple X. And actually, if it were triple X, it would die. So sometimes in the non-disjunction, just the chromosome simply gets lost. Okay? It doesn't actually have to go to the other cell. All right? So this can happen. So you'll get an XO cell, but this cell will behave as a male cell. And that's because the sex determination mechanism is cell autonomous. Each cell decides for itself. It flies, whether it's male or female. This does not apply to humans. Okay? So it decides for itself whether it's male or female. So if this happens early in the development of an embryo, so fly embryos is dividing nuclei, actually. Okay? And if it divides, so we're doing 2 different colors here. So the orange ones are going to be the ones that are XX, and the white ones are XO, males. And so you'll get an animal, an embryo that's a mosaic of male and female. Okay? And when that turns into an adult fly, so some of you in the lab have looked at male and female flies, so you know one of the differences is the abdomen has different patterns of pigmentation. So you can get an animal that's a mosaic. Part of its abdomen has male pigmentation, and part of its abdomen has female pigmentation. If you're out on the wing, you can't necessarily tell because the wing shape and everything is the same in males and females. Okay? So you would get a gigantomorph fly that's part male and part female. Okay? But this can also happen then with a ZW system, but in a slightly different way. So in this case, you would have a ZZ male, and you would lose a sex chromosome, and you'd end up with a Z0, a Z nothing, right, female cell in an otherwise male background. Okay? And so then this can happen, right? And one of the types, the 2 of the types of animals that have this mechanism of sex determination with the ZW system are birds and butterflies, right? And so many butterflies are actually sexually dimorphic. The males and females don't look the same. So often females are cryptically colored, okay, and males are very brilliantly colored. And the idea is that the females play it safe, they wait, the males are very brightly colored, they fight with each other, they get eaten by predators, and the females wait to see whatever pretty males are left around at the end of 2 weeks, and they mate with them because that's a proxy for good genetic health, right? And then in this case, the females are actually mimic of poisonous butterflies, but they only mate with these bright yellow males, and so you end up with this usual problem that the males really suffer at the hands of females like this, right? But you can get gigantomorphs. So what would they look like if that happened? So if you have this, and in butterflies are very spectacular, you get butterflies that look like this, okay? So this butterfly is half male and half female. Okay? And so one way that this may have occurred, and we'll talk a little bit more in a minute or later, is that is by this non-disjunction in mitosis. And so now you have 2 different genotypes as far as sex chromosomes go. And it doesn't have to occur just half and half. They can be mosaic like this. So in this case, this wing is these 2 wings, the forewing and hindwing on this side are mosaic of male and female, whereas it's all male on this side. Okay? And these happen with some frequency. So again, a normal male and female of this species, these are 2 what we call bilateral gynaetomorphs, right? Here's one where only this wing is male, and then these are all mosaic examples where there's complicated patterns of them being male and female. It also happens to birds. Okay, so here are cardinals. Okay, a male and a female cardinal, right? So that's the female, pretty drably colored and larger than the brightly colored male. And you can get cardinals like this, okay, that are half male and half female. Here's another example. Okay, and here's an example in chickens. Okay, so these are exceedingly rare, but they do happen. Okay, and so I'm going to leave you with this question: why can't this happen in humans? Okay.